Welcome to ConnectCon, presented by FTC Team 9794, Wizards.exe. Join us this week on our magical journey each day from 10 to 11 a.m. and 2 to 3 p.m. Eastern Time, where you can hear from STEM professionals from Google, APL, iRobot, and more. My name is Miriam Sinton-Remus, and I will be your host for this session. Please use the chat for any questions related to the presentation, and we'll address them at the end. Today, we're honored to have Dr. Erik Andersen speak about how a tiny worm can teach us about ourselves. Dr. Andersen is a biologist at Northwestern University. He likes studying worms so much that he even has his family collect them when on vacation. We will be talking about how modern genetics research is done at the university level. Then he will describe how his research group uses genetics of worms to understand drug responses, growth, and evolution. Thank you, Dr. Andersen, for taking the time to present at ConnectCon.
Great. Thank you, Miriam. Let me share my screen. Can you see it? Okay. All right, great. Thank you, Miriam, and thank you to the Wizards for inviting me to present today at ConnectCon. I'm excited to tell you, like Miriam said, about some adventures in biomedical science and what this tiny little worm can teach us about ourselves. So I've divided the talk that I'm going to give today into 5 different parts. And after each of the parts, I'm happy to take questions, and Miriam will be watching for any questions that come up on the chat. So feel free as I transition to each of those to write in any questions you might have. So at first, I'm going to tell you a little bit about my career path. So how did I come to be a professor at Northwestern and to be interested in genetics? And then I'll give a little bit of an introduction to genetics, and from there go to tell you about the power of model organisms and how we learn about human biology by looking at them. And then I'll turn to a description of my favorite model organism, Caenorhabditis elegans, this nematode roundworm. And the last bit, I'll tell you about what my lab does at Northwestern to understand natural differences or natural diversity across populations, to understand, again, how we are all different from each other. So in this first part, I'll tell you a little bit about my career path. So I grew up in Ohio and wanted to be a writer up until about middle school time. And in 6th grade, in our science class, we read an article about corn genetics. And specifically, it was using some genes that are present within a type of bacteria, and then moving those genes from the bacteria into the corn to be able to fight against insects and other things that might eat the corn. And at that point in life, I completely changed my direction from being a writer, and I thought, this is the coolest thing. I want to work on genetics. And became the obsession of my life to be able to run my own lab to understand these types of things. A couple of years later, after, again, naively thinking about genetics and what I wanted to do, I really love swimming and scuba diving, and I wanted to essentially make people have gills, because it's hard to hold your breath all the time. And I started trying to figure out how I can get involved in making people with gills. Luckily for all of you and for the rest of the world, a couple of years later, I began to realize that there's a whole lot more to genetics and to development than what I thought was going on during my time in middle school. So in high school, I've essentially dedicated myself to trying to get more involved in different aspects of genetics. And that brought me to Dr. Huntington Willard's lab at Case Western Reserve University, who's in the genetics department at that time. And I got to see a real lab in action and thought to myself, this is exactly what I want to do. So I decided to go to Stanford University for my undergraduate. Again, it was one of these places where I knew I could focus on research early on and spend my time learning more genetics along the way. I joined the lab of Dr. Matthew Scott, and his lab works on fruit fly genetics. And while I was in his lab for 4 years, I worked on how the fruit fly heart comes together, and essentially the same genes that make a heart in fruit flies are the same genes that make a heart in humans. So we can learn about the process of heart development in humans by looking at flies. So it was the first example I had for how we can take the similar genes across organisms to learn about biology within us. I then decided to go to graduate school at MIT, where I joined the lab of Dr. Robert Horvitz. And in Bob's lab, I then first learned about worm genetics and focusing on this Caenorhabditis elegans that I told you about, and very quickly learned that this is exactly the organism that I've been looking for and thinking about for a long period of time. And it really offers a powerful tool to be able to answer some questions about inheritance and about what makes populations that I've been thinking about for a long time. So after your graduate school, if you want to become a professor, you have to go and do a postdoctoral research. So essentially, after you get your doctorate, you do additional research. I chose to go to Princeton and join the lab of Dr. Leonid Krugliak. And in his lab, I specifically went there to learn the skills of computational biology, statistics, and programming, so that I can take those aspects of quantitative sciences and start to apply them to the worm genetics that I've been learning over the last 8 years of graduate school. And this then led in 2013 to having my own lab at Northwestern. So it takes about 17 years after high school to essentially get all the training and all the skills to be able to become a professor. And I wanted to kind of go through that just to give an example of this kind of academic career path and what happens along the way. Miriam, are there any questions at this point? Oh, you're muted, Miriam. Oh, none. Okay, got it. All right, excellent. I will continue and give you a little bit of a brief introduction to genetics. Okay, so genetics is the study of inheritance. An example that you might know well, or at least believe to be cuddly and cute, and that is dogs in this example. So let's say that you have a poodle mom and you do a cross with a Labrador dad. And from this cross, we're going to get this cute little Labradoodle puppy. So this puppy is a mixture between the two parents. And this is what's been observed in genetics for a long period of time. You take two parents that might be different from each other, they're crossed, and all the offspring, all the babies, all the puppies, whatever it might be, are a mixture of each of those specific parents. So in this case here, I'll just show a few examples. So the poodle mom, she has a small body. She's got a black fur color, and she has curly fur. Whereas by contrast, the Labrador dad has a big body, has yellow color, and has straight fur. So a mixing of those different traits, or those different parts of where these dogs, or how these dogs look, come together to make this Labradoodle puppy. So this puppy gets the big body from his or her dad, gets the brown color as a mixture of both mom and dad, of the black and the yellow, and gets curly fur like the mother, in this case, the mom. So this is what essentially gives us the basic rules of inheritance. And inheritance is controlled by genes. And genes are these basic physical and functional units of inheritance. In our Labradoodle cross that I'm showing you here, where we cross the poodle mom with the Labrador dad to be able to get the Labradoodle, all three dogs have a gene that controls their fur. And that gene has different types. Either it's straight or it's curly. But the two parents, they have a difference in this particular gene, and that's what makes them have a difference in their fur. And we call this difference in the gene an allele, okay? So you have to have a gene to be able to make this fur, but then differences in that gene, these alleles are what give you differences in your type of fur. So you can think of it like the flavor of an ice cream, right? So we have vanilla or chocolate are different types of ice cream, and just like alleles are different types of a gene. So the puppy gets both of these alleles, one from mom and one from dad, but only has curly fur because one of the alleles ends up being a lot stronger than the other one, and hence it's the one that we see in our Labradoodle puppy. So what are genes, and how do we find them? And this is where genetics comes into play. So genes are encoded in the genome of all organisms. So the genome is made up of DNA, this deoxyribonucleic acid, and it has 4 different letters, this A, T, C, or G. It stands for adenine, thymine, cytosine, and guanine. And you can think of the genome as the collection of all the genes, all these different letters that come together, and it's the instruction manual for life. So every cell in our body has this instruction manual, half of it comes from mom, the other half comes from dad, and it's what makes us who we are. So to follow our dog example here, again, alleles are these differences in the same gene, this fur gene, right? And we know that the genome of the different dogs might have changes in it. They have alleles, and those alleles are what make you have a difference in, let's say, the fur or the curly or straight fur. So in this example I'm showing you here, the poodle mother, if you just look at the list of A's, T's, C's, and G's in the genome, there might be one specific change. So for example, this cytosine at this location here. And if you have a cytosine at this location in this gene, that allele makes you have this curly hair. Whereas by contrast, if you look at the yellow dad, same sequence, same gene, but then a difference in that gene. Again, now there's a T, and that T is what makes this dog have the straight hair. So we want to look through all the genome to be able to find this particular fur gene. I made up this example to be able to illustrate this, but that's difficult to do. We can't just look at all these A's, C's, T's, and G's and tell you exactly where the fur gene is or what the fur gene might do or where or how it acts. So we need genetics to be able to figure out where this gene is in the genome and then to be able to do experiments to figure out what it does. So if we just look at a genome, again, it's this very long string. It has billions of letters in it. And the genes aren't labeled. We can't just go there and read it like we'd read a book to be able to know what kind of fur this dog might have, for example. So in humans now, moving to us, we've got 20,000 genes. And all those different genes are found somewhere within these 3 billion letters that make up our genome. And I'm just showing you some of this genome sequence here to emphasize that patterns are difficult to recognize. And you don't just know where to find these genes. And we often don't know what to do with them. Maybe we could find them. We don't know how they work. So again, this is what we do as geneticists. We look for these genes and we try to figure out what they do. So in many cases, we want to find these genes that specifically cause disease because we want to understand how to treat that disease. So different people have alleles of a gene that might cause diseases like cancer or diabetes or any others. And looking for these genes in the genome is like looking for a needle in a haystack. If we can find these particular genes, then we might be able to predict whether a person's going to get a different type of disease. We can also, if you learn about what the genes do and how they work, we might be able to make drugs to help fix what's going wrong or what's happened with that particular gene's function. So to do this, we need genetic crosses. We need to be able to cross, let's say, a curly and a straight fur dog. We need to be able to sequence the genomes to find out all those millions to billions to tens of billions of letters that make up that particular genome. And lastly, we need some of the new technologies that are around today for genome editing, like maybe this CRISPR-Cas9 that many of you have probably heard about, to essentially find these genes and to figure out exactly what they do. So let me go through this example again for humans. So human genetics is very difficult. Everything I've just described is not something we can do easily. So for moral reasons, we can't and we shouldn't tell people with whom they should make babies. So we can't really control crosses like we did with the Labradoodles. And we know that this genome is very large, right? It's got 3 billion different letters that are a part of it. So it's very expensive to sequence and to find all the different alleles that a certain person might have. Also, humans have a long generation time. It takes a while until each generation makes the next round of babies. So what would a scientist want to do, or how would a scientist want to wait 25 years for one cross to happen? And also, humans have very few offspring, right? In the United States, the average family has between 1 and 2 children. So if you wanted to understand more about how inheritance works, you don't have that many examples of that process going on. And as we've heard about the controversy from China a couple of years ago, genome editing of humans is prohibited. We need to learn a lot more about what the technology, how it works, and how to be able to do it safely before it'll ever be allowed for us to figure out what little changes, what little alleles in our genome cause specific differences in, let's say, disease or in other attributes of us. So how do we actually do this in genetics, and how do we get to find these particular disease genes? Okay, so that's the end of the intro genetics talk. Are there any questions yet, Miriam?
Yeah, so on the dog slide you showed, why is the fur a mixture and what happens to the dominant and recessive genes?
Okay. So why is the fur color a mixture? So there are some genes, so I said before that there are some genes that have alleles that might cause, as you just asked in the question, a dominant or a recessive phenotype. Or as I said in my talk a few minutes ago, some are just more powerful than others, and the more powerful one might be the dominant one. And you might not see the less powerful one or the recessive one. And so when those two from the mom and dad come together, you only see the most powerful one for some traits, let's say like the curly or straight fur. But there are others where there might be more than one gene, or there could be one gene, but there's not just one powerful type, one dominant type. There might be some where the alleles actually both contribute to it equally. And when that happens, you get this mixture where different colors can come together to in a way to make an intermediate color.
Okay, thank you for that answer. And do plant genes use the same chemicals as animal genes?
Do plants? Yes. So I should have said, and that's a very good question. Thank you. That the A, C, T, and G, these nucleotides of deoxyribonucleic acid that make up DNA, they are the same for all organisms on Earth. So one of the great, probably basal proof of evolution is that we are all related to each other based upon the fact that our DNA is made up in the same way from organisms as simple as bacteria all the way up to pine trees and elephants and whatever it might be. So plants, animals, bacteria, fungi, all of them use the same chemicals that make up DNA.
Okay, and the last question for this section, can you use genes to study and try to find a solution for COVID-19?
A good timely question. So yes, there have been a series of publications over the last 6 months, specifically in the last few months, where geneticists have done searches for genes that can change the way that SARS-CoV-2 or the virus that causes COVID-19, how that virus works within human cells. And there have been some interesting avenues for potential treatments that are out there. But it does take time to go from just the gene discovery to figuring out what the gene does and then how you can make a drug to impact the function of that gene. So we're at the early stages of this, but genetics has definitely been a major part of trying to understand more about how we can control this virus. All right, last question, right, Miriam? Okay. Excellent. So thank you all. Those are wonderful questions. I will continue to take them. So next, I want to explain how much of the project's progress in genetics has been facilitated by the use of different organisms that share their different biological processes with humans. And you can think of these organisms as kind of little humans, but we have the ability to do experiments with them in the laboratory. So the first of which is this baker's yeast that's known as Saccharomyces cerevisiae. It's used to make bread and beer and wine. So we use it all the time in different settings. We eat a lot of it. But there are also thousands of different laboratories across the world that use it as a model to be able to understand humans. We'll talk a lot about the roundworm nematode, Caenorhabditis elegans. I'll show you some examples of the fruit fly, Drosophila melanogaster. And the house mouse, Mus musculus, is also a great laboratory species because it's very close to humans in that we are both mammals, so it shares many of the same processes. So I'll take a look and compare them, each of these, to humans to be able to make these examples. So on the left here, I have the attributes of humans as a comparison, and on the right here, I'll talk about all those the four different species that we go through. And I'll break them up into the different attributes that make them favorable for understanding human biology. So yeast, as you can see here, have a very short generation time, right? Humans take 25 years to make that next generation, whereas yeast just take 90 minutes. So that makes them really easy to grow and to perform genetic crosses. I mean, you can just imagine how many different experiments you can do in the course of your average day, whereas you can never imagine anything like that with humans. They also have a very small genome. It's only about 15 million letters long. So it's much easier for us to figure out where those genes might be and what they do. They have fewer genes. And yeast and humans diverged from a recent common ancestor. So the last time that humans and yeast once shared their genetic information was probably over 1 billion years ago. So although it was that long ago, still about 23% of the genes you find in your typical yeast are the same as what you'd find in a human. So if there are basic cellular processes we want to understand, yeast is a great organism to use to understand those. And if they're shared with humans, it gives us even more power to know what these genes might be doing. And because of these pretty amazing attributes, there have been 5 different Nobel Prizes awarded just for fundamental discoveries using yeast. So for fruit flies now, this is a powerful model for genetics, and it's been that way for over 100 years now. So they take about 2 weeks for their generation time, so a lot slower than yeast, but definitely nothing close to humans. Crosses are very easy to do, and it's an animal, so it has many of the same organ systems and tissues that we do as people. And flies and humans, the last time that they shared this common ancestor was about 540 million years ago. So because they're much closer to humans, about 60% of the genes and there are 12,000 genes in Drosophila, about 60% of those are the same between Drosophila or fruit flies and humans. And genome editing is very easy to do. I should have said for yeast that genome editing is like this one of the places they've been doing it for over a decade. And in flies, it's so easy that there are actually companies out there where you can just go on the internet and you can order whatever edit to the genome you might want, and they'll deliver the flies within a month or 2. So it's something easy to do, this kind of fundamental research. Now we'll turn to the mouse. So again, like Drosophila, almost 100 years ago, researchers started to focus on this particular species. They're also easy to grow within the lab, although now it takes a couple of months to be able to go and get a next generation. So crosses are much, much slower, and it's more difficult to be able to find genes in this species. But mice and humans were both mammals. They diverged from humans maybe 80 million years ago. So that means that a lot more of the genes are going to be very similar between mouse and human, allowing us to understand a lot more about the biology that might relate to a human by looking at mice. And genome editing, this is one of the places where many different aspects of genome editing were first tested. There was actually a Nobel Prize granted for doing some of the first editing of a mammalian genome. And there have been 3 additional Nobel Prizes, so a total of 4 Nobel Prizes granted for research on the mouse. And I should say for Drosophila, there have been 6 different Nobel Prizes granted for basic research on Drosophila. So these organisms, although they may look very different than us, they are essentially the ways that we have been able to understand what genes are and what they do. And it's led to many fundamental aspects of our own biology by looking at them. So next I'll turn to Caenorhabditis elegans, or this roundworm. So it's a relative newcomer to the field of model organisms and the kind of hall of fame of organisms we use in the lab most often. So research on C. elegans started mostly within the 1970s. And this species has a very short generation time. It's only 3 or 4 days. So we can do genetic crosses quickly, especially for an animal. Its genome is relatively small, and we have the same number of genes as humans between a nematode and the humans. The most recent common ancestor, like flies, this case is about 600 million years ago, and 65% of the genes for that reason are pretty similar between nematodes and humans. And genome editing was worked out almost a decade ago now, so it's very easy for us to change any particular part of the genome in whatever way we wish. So it makes it into a powerful system to be able to understand biology. Okay, so those are for the model organisms. And before I turn to talk more about C. elegans, or this worm, and how much it can teach us about ourselves, Miriam, do you have any questions about the different model organisms?
Yes, so we have one question. How long does it take to identify one person's genome?
For a human, yeah. So it really depends on the technology that we use. So I should say that there are different technologies out there. Some of them have what we call short reads, where they take very small sequences of the human genome or of any genome, and then they sequence billions and billions of them and put them all together to figure out what that genome is supposed to be. And that takes about 4 or 5 days to do a human genome. And so it's a relatively quick technique. But because you've chopped up the genome into these billions of little parts, there end up being mistakes, and we end up missing some of the alleles that can cause differences in who we are. So there are new technologies out there that take very, very long reads. Now these are 10,000 or thousands of letters long. Those are a little bit slower, maybe a week or so to be able to get the data, but you're able to put them together to find more differences across genomes more easily. So I should emphasize that the speed of getting a genome sequence is not a huge factor for us anymore. So over the last, even let's say 5 years, it has been a kind of an amazing growth in the rapidity, essentially how quickly we can get this type of data. But it's what we do with those data that takes the large amount of time. So as I'm emphasizing here, so in humans, we might be able to determine the genomes easily and get all those letters all lined up in a way that we might be able to understand them, but we still can't read it yet. We don't know where to look, and we don't know, even if we could find those genes, we don't know exactly what they do, which is why we turn to these different other organisms to be able to learn about them. Any other questions, Miriam?
No.
Okay, excellent questions. Please keep these up. Thank you very much. So I'm going to turn my attention now to explain again what this little tiny worm can teach us about ourselves. So C. elegans is this powerful tool for genetics. I've told you about its fast generation time, its number of genes, but I want to go through a couple other favorable attributes that make it so we can do a large number of experiments with them. So these worms are cheap and they're very easy to store. So I'll show you pictures later when I show my lab, but we can grow them on little agar plates in the lab, and worms like to eat bacteria, so it's very easy to grow up a food source for them. And in my lab, in a very small incubator, maybe the size of your refrigerator at home, we can store tens of thousands to hundreds of thousands of different nematodes. So it's easy for us to be able to keep a large number of them and to get them going in our lab. And we can easily control the environment. We know exactly what to feed them. We know how to control the temperature very easily. We can even grow them in liquid, as I'll show you a little bit later. And one of the big advantages of C. elegans is that we can actually freeze them. We can put them in a very cold freezer or under liquid nitrogen, and they will stay frozen for as long as we want. And then we can thaw them later, and they come back to life again. So it's unlike most organisms. We can't freeze ourselves. We can't freeze mice, Drosophila. You can freeze yeast. But this is the only animal we know about where you can take it and put it into a freezer and save it forever. So the cool thing about what we can do in my lab and lots of other worm labs is we can study these genomes and we can look at individuals, but over time, they don't change. They're a stable resource that we have available to us all the time. And I've already explained to you in the expansive genetic toolkit here, I'm mostly talking about CRISPR-Cas9. So we have the ability to edit the genome. We can add in genes wherever we like, given you can inject foreign pieces of DNA into the worm. And I'll talk about that with one of the Nobel Prizes in a minute. And as I said, 65% of the genes and pathways are conserved between C. elegans and humans. So we can learn a lot about human biology by the overlap in their genes. And we've got a nice small genome, right? It's 100 million letters long, which seems like a lot, but compared to humans, it's much more manageable. But the really cool thing is this organism has been the basis for a lot of what we know about the human genome. So C. elegans was the first animal genome to be completed. And it was the first one to be completed because all the researchers that were essentially working out the tools to do the human genome, they did it on C. elegans first. So we had access as C. elegans researchers to a genome very early on in the process, back in 1998. And then in addition to that, we know a lot about how genes get turned on, where they are, where they start and stop, all because it's been a long time we've been able to have this sequence and to be able to do basic research on how it functions. So let me turn and I want to essentially show you that these different attributes are powerful and that thousands of different researchers work on this tiny worm for these reasons, and that we've had 3 Nobel Prizes awarded for research in C. elegans. So I want to kind of go through a few of those examples just to show you what we've learned from this tiny worm. So in 2002, the first Nobel Prize for C. elegans was awarded to Sidney Brenner, Bob Horvitz, and John Sulston. And Sidney Brenner is considered the father of the worm. So he was the one who brought this particular organism into its kind of general use right now, started up one of the first large lab operations to be able to understand how it works. And he chose it because it had this fast generation time and genetics was easy. And then two of his postdoctoral fellows, Bob Horvitz and John Solston, came and they wanted to figure out this thing called the cell lineage. And this is the process from a single cell dividing to make two cells, those two cells dividing to make 4, and so on up until the 959 cells that make up a worm. And the cool thing about the worm is this cell lineage, these divisions I'm talking about, they're the same from worm to worm. So these two guys, they went into a cold room and they watched from that single cell as it divides to make 2. You can see 2 cells here. And those 2 cells then divide to make 4, or 4 make the 8, and so on. And they followed every one of those divisions and they made a map. And that map is what we call the cell lineage. And from that, they were able to then look for mutants that break the process of this map. And those discoveries, once they figured out what genes were broken, that allowed them to be able to determine different pathways for how cells can talk to each other. So one cell, another cell might be different because one cell will tell the other one to be different from itself. Those fundamental processes were discovered in research based upon the C. elegans lineage. And a lot of these pathways where cells talk to each other, they're the exact same pathways that are at play in cancer, and they go bad when we get cancer. So the basic genetics of what genes are involved in the process, and when they break, they cause cancer, came from the studies of C. elegans and using this particular cell lineage that Horvitz and Solston defined. And along the way, they also discovered cell death, which is this process where certain cells are told genetically to kill themselves. And this happens in development. Again, it's something that goes wrong in cancer. So in normal development, I mean, all of us are when we're just before we're born, we actually have webbed fingers. And the process of cell death is what happens to essentially the cells between our fingers all die so that we have separated digits when we're born. So this was, again, fundamental biology to be able to understand how cells talk to each other. And the next Nobel Prize came in 2006, and it was awarded to Andy Fire and Craig Mello. And they discovered this process called RNA interference. And this one's more molecular for me to go to, so I'll skip many of the details. But essentially, it took them a long time when they made a basic observation that when RNA enters a cell, that RNA, if it's similar to a normal gene, it'll turn off that gene. And it seemed like a crazy thing at the time. And they went on to show that not only how it works within the worm, but then that it's not just worms, it's every animal that has this particular process, and plants as well. And that now this is one new therapeutic technique that's being used to turn off disease genes in certain types of diseases, and it's becoming more and more at play. And this came from studies in C. elegans. And the last Nobel Prize awarded so far was in 2008, and this is to Marty Chalfie, Asuma Shimomura, and Roger Chen. And they discovered essentially this protein that's present in jellyfish, and we call it green fluorescent protein. So when you shine a light on jellyfish, they glow green. And what Marty Chalfie did was he took that gene after Asuma Shimomura found it and injected it into C. elegans. And in the process, he was able to make C. elegans glow just like a jellyfish might glow. And here's just a video of showing how you can take C. elegans that are glowing green like the jellyfish will, and you can follow them moving around in real time. And it's not just a cool way to make glowing worms, but it's a good way to label any organism of interest. So this has been done on chimpanzees and cats and yeast and flies and everything. So we can use this as a tool to understand where proteins are and also where specific cells are within an organism. And it's because of this, it's an amazing tool for biologists. And again, this is a natural protein found within jellyfish. Shimomura found the gene for that particular protein, and this is not a gene that C. elegans or humans or anyone has. So we're able to use genetic engineering to put that gene into any organism to make it glow. And the cool thing is, because it's just a basic protein that glows green on its own, you can even label other proteins inside cells to figure out where those proteins go or what they might do. So it's a very powerful tool available to biologists. And the first application was on C. elegans in Marty Chalfie's lab, where he specifically looked at different parts of the worm brain that are required for recognizing touch. Okay, so Any questions about the kind of basics of C. elegans and how we do that, Miriam?
Yes. So does the formation of ice crystals not affect C. elegans when they're frozen, or are they frozen in a special way to prevent crystals from forming?
That is a fantastic question. I love these questions. Thank you. So yes, all cells get damaged by ice crystals. So what we do in C. elegans is similar to what you can do for many if you want to freeze bacteria or yeast or other organisms. And that is you change the composition of the media. So there's a little bit more glycerol and some other chemicals that can protect the cell membranes from these ice crystals. Ice crystals they're smaller. So because they're not large, they don't damage the different cell membranes or the outsides of the cells. And also John Sultan, actually, as I told you, with the Nobel Prize that he received for doing the cell lineage, he was actually the guy who figured out how to freeze worms. And the big trick that he found was that you have to freeze them slowly. So not only do you change the mixture of the solution that the worms are in, but you put them inside these special containers where as the temperature goes down to freezing temperatures, it does it at a much slower level, and those ice crystals are then much smaller.
Okay, next question. Can we use proteins to differentiate cancer cells?
Can we use proteins? Yes. So absolutely. So cancers have a variety of different genes or pathways that are turned on, and those genes and pathways, when turned on, make different proteins. And those proteins that they make sometimes are not normal for that particular cell. So you can tell that you have a cancer cell when a different protein is produced than what you would normally see in a particular tissue or a different part of a person, and so on. And a lot of I should also say that not just proteins, but how proteins are changed. So within our cells, a protein is not just one type. There actually are little bits that are added onto it, and those little bits our body can read to be able to say as a message to do something. So we can essentially use those little bits, those little messages, to predict as we measure them whether that cell or that tissue or that person might have cancer within them. And those little bits that are added on, and for the actual protein themselves, a lot of them, or at least 3 of the 6 pathways that we know cause cancer, came from the studies in C. elegans and also in parallel with Drosophila. So in the early 1990s, all of our knowledge about these genes and proteins that contribute to cancer kind of exploded based upon the fact that C. elegans and Drosophila researchers started to see similar genes within humans because more human sequences were coming out around the early to mid 90s.
Okay, and can the concept of freezing worms help save endangered species?
Another good question. I wish, yes. So it ends up that you can freeze some embryos from mammals and some other endangered species. They don't survive the freezing process perfectly in all cases, so it's not super efficient. But that is one thing that they are trying to do. It's easier to freeze single cells and specifically single mammalian cells, and then using essentially genetic engineering techniques where you can remove some parts of the cell and put it into another cell, you might be able to clone and make that species again. That's also being talked about, but at this point it's experimental and it hasn't been fully solved. So yeah, freezing embryos is possible. So you can freeze, let's say mouse sperm, you can freeze and save it, but you can't save the mouse embryo. So that's typically done if you want to at least save what the dad's genetic information might be. So it is an active area of research. And there are people in C. elegans that are working on different aspects of how you can slow down crystal formation in the way I just explained, and trying to make that more efficient in C. elegans and hoping that might work in mammals as well. And there's some suggestions that it could work that way.
Okay, and last question. Can you make fluorescent proteins glow differently based on the chemical environment?
Yes, and that's a great question. Thank you. So Roger Chen is on that Nobel Prize because that's exactly what he did. He changed that jellyfish protein to make it glow different colors. And then he also made it responsive to different chemicals. So you can change the pH, the acid or neutral environment or a basic environment of a cell, and you can make it glow different colors depending on that. He made it sensitive to different types of ions like calcium. And that has, the major contribution that he has for the Nobel Prize is the fact that this now, not just GFP, this green fluorescent protein, but all the other things that are possible that he's made just by simple mutations to this one jellyfish protein. Great questions, thank you. All right, so I'm going to turn to tell you a little bit about what my lab does and be able to understand genetic differences across populations and to understand more about how we as humans are different from each other. So each of our genetic differences make us unique. So in this picture here, you can see, and you know, if it weren't the time of COVID and we were in large groups, you'd be able to see there's a lot of diversity across the human population. We all know we look different from each other, but not only do we look different from each other, we also behave differently. We respond to drugs differently or different treatments, and we also have different propensities for getting certain diseases. So if we can understand all the genetic differences across a population, especially for certain diseases, we might be able to predict who would get sick or who might look or behave a particular way. And if we can do that, we also might be able to know who would respond differently to a particular drug or treatment. And again, having those predictions can make health even more efficient if we know who will be responsive and who will not be responsive to a particular drug. So for example, let's take chemotherapeutics. So etoposide is a common chemotherapeutic, and we use it to treat and kill cancer cells. But some people across the human population, they respond more than others to etoposide. So they get more of these side effects, they might get sick more, and because of that, it doesn't treat the cancer as effectively because the dose that makes it to the cancer is not as high as what makes this person sick. Whereas on the other hand, some people respond to drugs a lot less than others. So it's going to take a much higher concentration or a higher dose of etoposide to be able to actually treat that person's cancer. So if we're able to predict individual differences in etoposide response, then we can treat cancers more effectively. So to investigate how individuals differ in responses, we need three different things to know. We need different individuals to be able to see their genetic, we know that they have different alleles from each other. We need to be able to sequence genomes to find those different alleles. And then last, we need to be able to measure these differences in drug responses so that hopefully we can figure out what genes make them different from, or what alleles make them different from each other. So in my lab, we focus on C. elegans again, but we specifically look at populations of worms. And these worms are found all across the world. So my lab goes and collects them, and many of our friends and collaborators go and collect. And we get these different nematodes, and they're as different as you and I are from each other. So if we want to understand differences across a population of humans, we can use differences across a population of worms. So these worms come from different substrates. They come from rotting fruits, terrestrial isopods, from agricultural settings, or from compost heaps. Essentially, any place that's going to have rotting material and bacteria, you're going to find nematodes like C. elegans. So again, my lab goes out into the field to collect. Here we are on some strawberry field collecting rotting strawberries in Wisconsin to find nematodes on them. My lab often goes to Hawaii to be able to identify nematodes from those environments. Here I am on St. Lucia collecting nematodes that I'd found in one of the rainforests there. So you can do this very easily and around different parts of the world. So once we're all allowed to travel again, it would be really great if any of you in the audience right now are listening and are interested. I want you to go out there and collect nematodes as well. So you can go to my webpage here and read more about it, or you can send me a message on Twitter or email me, and I'm happy to talk about it. It's very easy to do. You just go and find, again, rotting leaves, rotting flowers, rotting palm nuts, rotting mushrooms, whatever it might be. And in these cases, again, you can just send them back to the lab and we'll identify the nematodes from them. Many middle school and high school students have done this for us. And it's really a great opportunity for all of you to contribute to kind of active science that's going on and to really participate in laboratory research. But this is how we get different individuals. We and people go out into the field and find these different worms. Then once they come into the laboratory, we freeze them. Again, we save them so we have them for a long period of time. But then we also need to sequence their genomes to find all the alleles and differences across these genomes. So we use different technologies to do it. Again, I talked about short read sequencing, so billions of little tiny fragments. That's what Illumina uses. PacBio and Oxford Nanopore technologies, they're the much larger fragments I was referring to. And again, all of these come together to be able to put together a genome. And oh, I should say Oxford Nanopore Technologies is really cool. They make sequencers that actually fit on your desktop. You plug them into your laptop or your normal computer, and you can sequence genomes sitting on your desk at home. It's an amazing technology. It's just come out in the last few years. But to do all of this, biologists need to have knowledge about programming. We should understand statistics. And ultimately, this comes together into the field of computational biology. So having the types of skills that many of you think, I don't want to be a biologist because I really am focused on math and other aspects. Most of biology now has a lot of programming and statistics as a part of it. And because of that, it's much more of a quantitative discipline than it ever used to be. So we collect worms from the wild, and we then bring them to the lab and sequence their genomes. And the next thing is to measure responses to a drug. So again, with etoposide, we know that etoposide prevents cell proliferation in humans. So we can use essentially this drug and throw it on our worms. And one or two different things about worms that are the same as the cell proliferation we might see in humans are that worms tend to make fewer offspring if they're exposed to etoposide, and their body size is smaller. So we just need a way to be able to measure the number of offspring and how big the worms are. And we want to do it for thousands of worms. So we can't just look at all of them. We have to do something better than that. So we take advantage of this device in my lab called a worm sorter. It's made by this company called Union Biometrica. And what we do is we can grow worms in liquid, not just on plates, but they can grow in liquid. They can eat the bacteria in liquid. And then this machine will suck up those worms, and it'll send them down a little tube, essentially. And when those worms pass through a laser, that it'll tell us how long that worm is, and also how optically dense or how thick that worm is. So each worm, we get a particular measurement of its length and its optical density or its thickness. And it's not just one worm. We can measure hundreds of worms per second as they go through this device to be able to give us really nice data about every worm and OTIS. So each of these little dots here, that's a worm that's gone through this particular device, and we'd measure these two attributes. And here's some real data. So this is a collection of, I don't know how many, a couple hundred worms, and they range in size from big worms to little worms here. And we have this type of population to say that this is either a healthy or an unhealthy population after exposure to a particular drug. So let's take etoposide, this chemotherapeutic I was telling you about. If we compare our no drug or our control situation, we get a nice growing population of worms. So they have large sizes, there's a large number of them. Whereas if we expose them to etoposide, they're all much smaller, right? Their length is smaller and their optical density or their thickness is smaller. So these worms are sick, and there also are fewer of them here, so their offspring production is down. And this gives us a way to be able to understand what the drug is doing to these worms. So we grow them in 96 well plates. So these are 96 little cultures, all of them filled with about 600 to 800 worms in each one. And then we can run them through this device to be able to measure across each of these 96 wells, what do the worms look like? Sometimes we get nice growing populations. Sometimes we get sick populations. And this tells us who responds more or who responds less to etoposide. And in my lab, we also have imaging platforms that work the same way. This is another type of robot, but this robot is a robotic microscope. So it goes along each of those 96 different cultures I told you about, and it takes a picture of each of them. And again, as humans, we'd have to go and look at all these and measure them, and that's difficult and tedious. So in my lab, we've written a variety of software and code to be able to identify the worms across this image, to be able to figure out where the lighting is best so we can count them most reliably, and to be able to understand the lengths of these worms, and also to look at them actually physically blow up and understand more about what this worm looks like in the presence of a chemotherapeutic. So to do these types of studies, you have to understand how to do robotics, how to put different machines together. Again, it takes lots of aspects of programming and statistics to be able to do it reliably. So all that comes together for us to be able to measure how worms respond differently to drugs. And in my lab, we've measured responses to a whole bunch of different drugs. So I'll tell you one example here today about etoposide, but I'll just generally mention some of the others later. So for etoposide, again, this chemotherapeutic that we take, we know that this drug binds to human topoisomerase 2. It also binds to other topoisomerase proteins that are out there. And I'll tell you a little bit about what topoisomerase does to tell you how etoposide is a poison of this particular protein. So in this video here, you're going to see twisted DNA put together, and two hands are going to come in. Those two hands are our topoisomerase. And topoisomerase's whole job is to break the DNA. Actually, it's going to take the energy of ATP, so these little discs. It's going to break the DNA into two pieces. It's It's going to untwist it, and it's going to put them back together again. And what etoposide does is it prevents topoisomerase from putting those two pieces of DNA back together again, and that breaks DNA. So that's why it works as a chemotherapeutic, because it leads to lots of breaks in the DNA, and that makes cells die. So we put etoposide on our worms. We tried to figure out who's responsive, who's not responsive. And without going into all the details, I'll just say that we found one single part of the genome that makes some worms respond and other worms not respond. And it ends up that that gene it encodes or makes topoisomerase 2. But specifically, it changes the way etoposide binds to or touches this topoisomerase 2. So one nucleotide or one base, one letter will make them bind more, and another base will make it bind less. And that's what makes you more or less responsive. If it binds more, you're more sensitive. If it binds less, you're less sensitive to this particular chemotherapeutic. But this is worms. We then went on to human cells. We used CRISPR-Cas9, just like we did in worms, but in these human cells, we edited them to make them from the sensitive version that's out there. The blue is the sensitive in these cases. And we added on and made some of them into the resistant version. The same allele that we find in C. elegans, we made into the human cells. And then we exposed them to either etoposide or to a control. And as these cells are given a drug, they start to grow if they have the resistant version, because now etoposide doesn't hurt them anymore. So we get a lot more human cells that survive with this particular allele than they do with the sensitive allele. So this tells us that worms can impact what we think about chemotherapeutics. So this is great. We can now take our worm knowledge, we can understand that, we can make predictions about how humans will respond to etoposide. And this is one of the major goals of my lab, and this etoposide is just one example. And over the years, we've done this for a series of other chemotherapeutics, heavy metals, and anthelmintics. So these are anti-nematode compounds. So when they overlap, when the genes are the same between C. elegans and humans, we have this opportunity to learn about it. And when they're not the same, we have the opportunity to learn about how evolution could have worked to make worms and humans different from each other and how we respond differently to these different drugs or toxins. So I hope that this has made it clear that we can use C. elegans populations to understand about human populations. I'm happy to take any questions about this natural diversity section now, Miriam.
Yeah, so we've had lots of questions. So the first question is, how common is it currently for doctors to use genetic information to decide on treatment plans for patients, and how effective is it?
That's a great question. Yeah, so there are over 100 different drugs that we take that now have a genetic test on the label. So there are genetics being used for whether you take that particular drug or not.
Okay, so the next question is, can we modify insect genes and introduce them as a new species to make the new species dominant?
There is a whole vein of genome editing to be able to do that type of approach. It's called gene drive approaches, if you want to look it up. But yeah, you can do that with almost any species. It also can be done with nematodes. It can be done with single-celled species as well. And that's one potential scary, but also possible mechanism to control populations.
And do you see a lot of diversity in the genomes from worms collected from different parts of the world, and how do you compare them?
Yeah, good question. So yes, we do. And we find that certain places, specifically the Pacific and Pacific Islands like Hawaii, have more genetic differences than worms that we find from Europe, the United States, Africa, and so on. And we sequence their genome to be able to compare them to each other, and then we run them through all of our different robots to be able to look at how different they are in drug responses and other attributes.
Okay, and the next question is, in order to measure hundreds of worms per second, how fast do the worms have to be in order to have shot through the worms shorter?
Oh yeah, I actually don't know the speed that they go through the worm sorter, but they go through very quickly. So it takes about 40 seconds to run those 600 to 800 worms through. I'm sorry, it takes about 25 seconds to run those worms through. So they're going through pretty quickly.
And why can't you use all sorts of model organisms to make a generalized statement?
Oh, you absolutely can. And it becomes more powerful for the more model organisms as you compare them to each other. So the more, whenever things are genes are the same or act the same way across more model organisms, it's more likely that humans will be the same way.
Okay, next question. How do worms react to caffeine?
Worms respond to caffeine. So caffeine is actually a pretty dangerous toxin for most cells. I know we all might drink it and have it as a part of a normal diet. So in small amounts, it's a stimulant, but in large amounts, it actually kills cells. So it actually kills worms at the levels we look at it.
And what is an example of worm diversity?
An example of worm diversity. So without talking about genome differences, an example would be like this etoposide response. Sometimes worms respond and they get sick and die. And in other cases, they don't respond and they're resistant. So we have many drug examples that are like that, but there are others too. Some like to wiggle more than others. Some like to swim faster or slower. But you can measure all those things with robots and computers, and that's what my lab does.
Okay, so that was the last question. If you have any more questions regarding the presentation, please email them to us and we will forward them to Dr. Andersen. ConnectCon continues tomorrow with our 2 sessions. In the morning, iRobot employees will be speaking about their career paths, and in the afternoon, Mr. Alan Nuttall will be speaking about his career at Google and Uber. If you would like to be informed about more events and videos by our team, please subscribe to our YouTube channel, follow us on Twitter, Instagram, and Facebook, and check out our website. Thank you, Dr. Andersen, for presenting at ConnectCon. Thank you, everyone, for attending the presentation today. Also, thank you to our alumni, Sarfik Vatnagar, Angeline Zing, and Nishan Oberoi for their help. See you tomorrow.
Thank you, everybody. This is great. Thank you.